And you already can see what she already introduced you about my topic, so I'll straightway go to my slides. So I'll talk about the introduction, what's GI cancer, its incidence in New Zealand, what the transporter protein I am interested, and the anti-cancer drug I'm working in this project, and which come my hypothesis, aim, and the results I finally got. I can share few of them, and finally what I've concluded with these results. So cancer, I am sure like everyone must have known what is a cancer. Cancer is a large group of disease characterized by uncontrolled growth and spreading of abnormal cells. And what is gastrointestinal cancer, which is also called GI cancer? That is a group of cancer affecting your digestive system. Pancreas, liver, small intestine, your colon, which is also known as colorectal cancer. So when we see its incidence in New Zealand, it is one of the major health issue for the New Zealanders, because I think around 30% of the New Zealanders are dying because of the cancer, and one of the major cause of hospitalization over here. By the Ministry of Health, it has been predicted like annually 3,000 New Zealanders are affected by GI cancers only. So you can say pretty much like it's one of the major health issue for the New Zealander. And there are like few treatment process for treating GI cancer. Everyone knows like for cancer now surgery along with many other therapy like chemotherapy, radiation, immunotherapy. For chemotherapy we use different kinds of anti-cancer drugs. Like sometimes cocktail of anti-cancer drugs, sometimes only particular anti-cancer drugs, and some of the chemotherapy process have also cured metastatic cancer like testicular, few cases of ovarian and lymphoma. And for my study, I am mainly looking for the platinum-based anti-cancer, because platinum-based chemotherapy is very effective. You must have known this guy. He's a former professional cyclist, and he has been diagnosed with testicular cancer earlier during his cycling career. And doctor already told him like his survival rate is only 20 to 50%, like it's quite low. And then he has been given cocktail of all the anti-cancer drugs along with platinum-based chemotherapy. And then after 6 to 7 months of chemotherapy, he has been declared as cancer-free. So you can say like platinum-based chemotherapy has saved his cycling career earlier during his earlier career time, not now. And the drug I'm interested is Oxaliplatin, which is third generation based platinum drug, which is approved by FDA for curing colorectal cancer and few of the lung, ovarian, and pancreatic and liver cancer. So they have a characteristic, they form inter and intra-strand DNA link, which cause disruption of the DNA and finally cause killing of the cancer cells. But in spite of having such a great anti-cancer killing property, the patient who respond to the oxaliplatin chemotherapy, they started showing drug resistance after 6 to 8 months the chemotherapy has been initiated. Like why? What can be the main reason? One of the reasons may be because of the expression of ABC transporter. So this ABC transporter, which is they use ATP as a source of energy to translocate substrate across the membrane against the concentration gradient. And the one of them I'm interested is MRP2. MRP2, which is a member of ABC transporter, and it involved in multidrug resistance, and it been encoded by ABCC2 gene in human, which is highly expressed in the apical part of the liver cells, which help in the biliary transport and some of the apical membrane of the proximal renal tubule, which helps in the excretion of the iron. So you can say that this gene is pretty much expressed in the organs which help in the absorption, distribution and excretion of the ions and substrates. So MRP2, they cause resistance of few of the anti-cancer drugs like cisplatin, vinblastine, and now I can't remember right now at this moment. And it also has been shown that it caused the efflux of the oxaliplatin anti-cancer drug. So what happened when chemotherapy is given to the patient? This transporter, they pump out most of the oxaliplatin from inside of the cell to the outside of the cell. That's how all these cells, they survive chemotherapy, because they're not receiving enough concentration of the drug inside the cancerous cell to kill those cells. So here is my hypothesis, like MRP2 contributes at the oxaliplatin resistance at the cellular level in this GI cancer, and if I remove this kind of transporter, it will reverse the drug resistance. So my main aims is to examine the cellular cytotoxicity of the oxaliplatin in GI cancer by removing this kind of transporter, and then to examine the platinum accumulation in these cells after removing the MRP2 transporter, and finally to find the correlationship between the oxaliplatin efficacy and the MRP2 expression. So for that, of course, I have to maintain cancer cell lines, otherwise I can't proceed. And then I will maintain the cancer cell lines where MRP2 gene has been silenced. For that, we'll be using RNA interference tool. It is very specific and with minor effect. And the RNA interference we are using is siRNA gene silencing technology. So, which is of different mechanism, but I don't have that much time to explain. So just remember, like control cancer cell lines and MRP2 silence cell line. So just for the convenience, just say MRP2 cell lines. And then I have to quantify the mRNA level of mRNA level in siRNA cell lines to see how the MRP2 gene has been expressed. So for that, we are using light cycler 480 PCR, which is an ideal combination of speed, accuracy, and versatility. It's a really good machine for to quantify the mRNA level of MRP2. And I have 12 genes of interest along with the reference gene. So when I measure the mRNA gene expression of this siMRP2 cell line against the control cancer cell line, the mRNA level has decreased by almost 50% for all the MRP2 knockdown cells. So I forgot to tell you before, I have three different siRNA sequence. So like for one cell line, they will be knockdown percentage will be different for those three different siMRP2 cell lines, and the knockdown percentage among them is almost 30 to 50%. That means the siRNA has successfully able to knock down MRP2 gene in these cell lines. So next will be, I have to see whether it's knocking down the other related genes or not, because I am not interested to knock down other genes which can affect the drug resistance or affect the transport of the oxaliplatin. So it has been shown there was no knocking down in other transporter or the other related genes, which can be say as negligible off-target effects. So second, I have to go to check the cellular activity of MRP2 transporter, like how the MRP2 is transporting the substrate across the membrane. So for that, I will be using flow cytometry. Flow cytometry, they detect fluorescence signal and then separate the cells of interest based on the light scattering property and the fluorescent property. So when we'll get the flow, when more amount of substrate will be effluxing or will be accumulating inside the cells, then the fluorescence will be more. More fluorescence, that means we'll get more signals. So generally we separate the cell based on the cell relative size, that's the forward light scattering, and the cell relative shape, that's the side light scattering. So accordingly we will get the cells, we'll separate our cells. And after that, it has been shown that at one time point with the inhibitor which inhibit the activity of MRP2, the fluorescence signal was 3-fold more than without the inhibitor of the MRP2, that's represented by blue line. And when at that time point period I have measured the SI-MRP2 cell lines cellular transport activity, and it has been shown they have been like accumulating more amount of substrate. That means the MRP2 genes have been knocked down, that's why they've been able to accumulate more substrate inside the cells. So the third one I went for to test the cytotoxicity activity, like how the oxaliplatin has been reacting inside this kind of knockdown kind cell, and for that I'll be using MTT assay. So with the MTT assay, it has been shown that the oxaliplatin concentration are killing more amount of cell, means the oxaliplatin sensitivity has been increased, which is still going on. That's why I couldn't able to show you the results. But it's like the mRNA sensitivity has been increased for the SI-MRP2 cell line, the knockdown cell lines, then the control cancer cell lines. And the last part, we have to calculate how much concentration of platinum has been accumulated inside the MRP2 knockdown cell lines. For that we will be using ICP-MS, which is inductively coupled plasma mass spectroscopy. It's an bioanalytical analysis, and thanks to Professor Mark McKeach, he has been allowing us to use this machine. And it is very like reliable and well validated machine for determining the platinum accumulation inside the cancerous cell. So what my conclusion will be at last, like RNA interference has successfully knocked down your MRP2 gene in our cancer cell lines, in GI cancer cell lines, and the knockdown percentage was 30 to 50% without any negligible effect. And after that, in this kind of cell line, the MRP2 expression, mRNA expression has been reduced by almost 50%, and this cell line has shown increase the substrate accumulation inside the cancer cell, and it's also shown increase the oxaliplatin sensitivity inside the cancer cell. So all this result can conclude that it may reverse the drug resistance in the platinum oxaliplatin chemotherapy. And at last, I would like to thank my supervisor, Dr. Yan Lee, Professor Jun Lu, Dr. Febres, and all my lab mates. Thank you all.
Thank you, Riya. It was a very informative presentation and energetic one.
Hopefully everyone must have understood, but that's
Indeed. So, any questions? It seems nobody have a question. I will take the place, and I wonder that about this platinum drug that you introduced it today for me for the first time. So is it important, what is the stage of the GI cancer that you are going to look at the effect of the drug when you are collecting your data?
Because we haven't gone through like which stage of the GI cancer. We are just the starting phase of this the experiment. So like how this drug will be affecting only the first stage of the GI cancer, but that will become after successfully we'll do these in vitro studies, like which stage is more effective. Then we have to go the bioanalytical analysis, like which metabolite of oxaliplatin is more effective for the GI cancer stage.
Okay. So that is really can matter the situation, you think?
Yeah.
Okay. Thank you.